So we finish out chapter 23 talking about some of the technological applications of bacteria and fungi. So basically how we are using bacteria and fungi for our benefit. This is separate from some of the ecological services that they provide, such as decomposition and nutrient recycling. So these are things that we use them for. Be a real quick list of some of the reasons that we love them so much. One is cheese. So if you happen to love cheese, you can thank some bacteria because they help to turn lactose into lactic acid, and that produces cheese as part of the cheese making process that has to happen. Fungi are also important. So if you happen to like stinky cheeses, like blue cheeses, fungi are important for that. The blue marbling that you see in a blue cheese is actually a fungi. So if that grosses you out, I'm sorry, I happen to love blue cheese and I'm not going to give it up. Other fungi are used to produce soy products such as tempeh, and they are also doing fermentation there. Soy sauce is made by fermentation as well. And then of course we've got the yeast Saccharomyces cerevisiae. It is used in bread making, so that lovely smell that you get from fresh bread when someone in your family cooks it or you walk into a bakery, that is the alcohol that is being released from the yeast fermenting, and then that alcohol is baked out. And all those lovely little bubbles are the carbon dioxide that are released as part of the fermentation process, or the alcohol that is being released from the fermentation process. Brewing is the same thing, fermentation of generally wheat and other products, other crops for making beer. And then of course winemaking is fermentation of yeast, all done by our friends, the Saccharomyces cerevisiae. In the chemical industry, we use bacteria for a variety of things. Basically, we use organisms such as E. coli to be workhorses for a variety of things that we need to produce. So for example, we can engineer bacteria to produce enzymes for us. We can use them to produce proteins such as insulin and vaccines, antibiotics, etc. This is generally done by genetically modifying an organism. One thing that we do quite frequently is use it to make pharmaceuticals. So one example is using E. coli, our friend the bacterium E. coli, to produce insulin. So initially insulin was produced by extracting it from a pig pancreas. So to get a little bit of insulin, they had to slaughter a lot of pigs. And so a couple decades ago, genetic engineering processes allow for inserting the human insulin gene into E. coli. And because the processes of transcription and translation are essentially the same in bacteria and in eukaryotes, and the genetic code is essentially the same, we can use E. coli to make those insulin products for us, extract them, and then use them for pharmaceuticals. So it's much cheaper. Many less pigs are dying in this process. 0 die in this process. So it's also a lot cleaner, better for pretty much everybody involved. A different sort of use is the use of a DNA polymerase taken from an organism called Thermus aquaticus. This enzyme is a high heat stable DNA polymerase, meaning that it can copy DNA molecules, and it is used for a process called polymerase chain reaction. In polymerase chain reaction, we're basically doing DNA replication in a test tube. So PCR is kind of the basis of pretty much all of our genetic technologies in a lab. All of our biotech, all of our GMO processes are going to depend on PCR. And in PCR, we can make a lot of copies of a little bit of DNA. So we can extract a little bit of DNA and make lots and lots of copies, which is how we would have produced this insulin gene that we're going to put into the E. coli. So Thermus aquaticus is an organism that can withstand really, really high temperatures that would denature most other enzymes. And so DNA polymerase is taken from this one because the process of PCR requires that we heat up the reaction up almost to 100 degrees Celsius, not quite, but enough to relax the DNA so that the DNA polymerase can get in there and copy it. And then finally, we use microorganisms for bioremediation. Bioremediation is where we use bacteria usually to break down organic compounds, to clean up our metal waste, absorb pollutants, etc. This is useful for wastewater treatment, contamination, oil spills, metal waste, all of that. The example here are organisms that break down hydrocarbons. So they can actually be seeded, S-E-E-D-E-D, into or on top of an oil layer from an oil rigger spill. And the bacteria will basically munch on those hydrocarbons, release carbon dioxide and water, and then the hydrocarbons, anything that's left kind of settles down to the bottom when those bacteria die. So they can be used for a variety of different things depending on their metabolic capabilities and the use that we need them for.